I've got the live warning on my Zoom, so it looks like it should be going now, which is fabulous.
Okay, cool. It doesn't look like it's live on my side yet, but that's great.
And I can see it on our Facebook page too, so.
Yeah, it says live for me on Zoom as well.
Okay.
Yeah.
Looks like it's still working on it. Okay, great.
Awesome.
Welcome, everybody.
Okay. Let's let in people.
Okay. You're going to do that?
Yes.
Okay.
Hey everybody, welcome to our Beaty at Home. We'll start in 2 minutes.
Hi, everybody. While you're waiting, please let us know what your favorite plant is, if you have one.
Only one?
For now.
Okay. So it's 1 o'clock, so we'll get started. So welcome everybody for our Beaty at Home. Today, we're very lucky to have Linda Jennings, who's the collections curator, Vascular Plants and Algae at the UBC Herbarium. She's going to tell us all about pressing and preserving spring plants. So before I hand it over to Linda, I'm just going to give you a bit of an introduction to the museum and to the session. So of course we've got Linda, and then my name is Kashifa. I'll be hosting, and Nancy Lees will be facilitating your questions in the chat and over at Facebook as well. I wanted to spend a couple minutes just talking about the Beaty Museum. So I wanted to do a land acknowledgement first. The Beaty Museum is on the traditional unceded and ancestral territory of the Musqueam people. And so you can see the flag of the Musqueam here on the screen. And I've also included a link to the First Peoples map of BC. So wherever you live across the province, it will help you find the languages spoken on the territory. Here we are on a map. So the Beaty Biodiversity Museum is here at UBC on the peninsula, and we have lots of other wonderful places on campus as well. We have the Pacific Museum of Earth, the Museum of Anthropology, the Botanical Garden, the Balkan Art Gallery, so lots of cool places to visit when it is safe to do so. This is what the museum looks like on the outside. We've got that huge blue whale skeleton, but we've actually got over 2.1 million specimens in the museum. So lots to see, including lots and lots of plants and plant-like things. Next to the museum is the Biodiversity Research Center. There are over 50 faculty, graduate students, postdoctoral researchers who are studying all aspects of biodiversity. And within the museum, we have 6 different collections. So you can see them represented here. And Linda, of course, is in the herbarium, which is represented here by a mushroom, even though fungi are not plants, but they're still part of the herbarium. So with that, I will hand it over to Linda. So I'll stop sharing. Here we go.
Hello, guys. So I am so glad you guys have joined us today. I am really excited to talk about pressing plants. So I normally do a workshop in the summer, and of course we can't be seeing each other face to face, but I really wanted to take this opportunity at Beaty at Home to see if we could try to share a couple of ideas. on how to press plants, and I've been starting this backyard biodiversity. So I've been making a few videos online on the Instagram website for Beaty, and it's just trying to introduce you to some of that backyard biodiversity you might be finding or something around your short walks around town. And then I'm also doing an Instagram with pressed plants, and that also is kind of giving you some more techniques. So that's going to be a little bit more focused on techniques that we're going to want to do with different kinds of plants that you might find, again, that you're pressing. So I'm going to do a short talk, and I totally welcome questions along the way. This is just going to be an introductory talk, just so I can show you a couple of the things that about the herbarium in general, and then get into that, some of the details of pressing. And then we're going to flip into actually pressing a few plants and giving you a few pointers on how to start out your own press collection. So the first thing I'm going to do is I'm going to share my screen and get to my PowerPoint presentation. So I'm Linda Jennings and Collections Curator. So I take care of the vasculars and the algae, but I have a love of fungi and bryophytes and lichens. So I want to start off by just saying this is a classic, beautiful example of a pressed specimen. This is Gerald Straley. He was our director and also in the botanical garden as a curator. And he, as you can see right there, he cut that base of this plant in half so that you could see the layers inside of this bulb that's at the base. He also spread out a lot of those flowers and then also cut those leaves up so that they could taxonomically look at it. So to be able to not only make a scientific specimen that's also beautiful, that is a talent. And Gerald Straley has that talent. And so I wanted to show you what I consider just a beautiful specimen that actually spans both of those worlds of scientific, but yet also quite beautiful. And of course, it's also called Spring Beauty. So what an amazing spring we've had, correct? There we go. So herbarium is where I work, but the plural is herbaria. And herbaria, we preserve a lot of biodiversity. So we preserve the algae and the fungal diversity and the plant diversity. And these are dried reference specimens, and they actually can date back to almost 450 years. So I'm going to try to see if I can get myself out of the way. Registered herbaria in the world. So we got 3,324. So almost any country you go to is going to have an herbarium. But there's an estimated 392 billion specimens in these repositories, and only 10% of them are digitized. So as we sit at home right now, it's been quite difficult because a lot of our specimens have still not been digitized, but hopefully that will change, and we'll get that funding to get even more of these specimens digitized so we can see more of this diversity that's sitting in these cupboards. The largest one is in Paris, and it's 800 million specimens, which is a heck of a lot of specimens in one place. But the oldest one is in Germany and Italy, and they're 451 and 450 years old. So one of the reasons I'm bringing this up is what's amazing about plant specimens is they're going to last as long as they're taken care of. And this is about the preservation, and this is my job, and this is what I do, because I know this is not just about the next 50 years. This isn't about the next 100 years. These specimens can last as long as they're taken care of. And so this particular collection has been taken care of quite well. And it's important. So it's important when you're making these specimens to realize that they really can have a longevity as long as they're taken care of and preserved. So in Canada, we have 123 registered herbariums. So there might be a lot of herbariums that are around that are people's personal collections, which is great. But there's also a registration for those collections that actually want to share and be able to make loans and ship those all around the world for research. So we are actually a very large collection, so 713,000 in our collection, which is quite strong. And but a lot of people always think, oh, we're the biggest, but actually the biggest is the Department of Agriculture. It makes a lot of sense. They were some of the first ones in Canada looking around, looking for things that were useful, looking for new invasives. And so they actually have the largest collection in Canada. The cutest one and the littlest one is in the Yukon, which is Bruce Bennett. So that one at the very bottom, I really appreciate Bruce Bennett. He's up in the Yukon, and this collection is actually registered. It's his personal collection, but it's registered. He actually sends this around the world for research, and it's in his basement. So you too could become a registered herbarium by the end of this. So to give a shout out to our initiator, our start of our whole herbarium is Mr. John Davidson. So this was the provincial herbarium before it became UBC Herbarium. And it was basically his thought processes and his work that actually got the first herbarium started here in the province. And so this was his office, where he actually has a lot of the herbarium specimens, but they were actually held up in upright books instead. So behind him is actually a lot of those herbarium specimens. We now lay them flat because it's a lot less difficult and hard on those specimens, and then house them in their cupboards. The way to think about herbariums is they're time machines. So we're really lucky right now because we're going to be able to go out and we're going to be able to see these berries and we're going to be able to see these flowers at this time of year. But of course, when winter comes, what if you're still working on that taxonomic work? What if you still needed to look at that level of detail? Well, this is where the plant specimens come in. And so you have these pressed, beautiful dried specimens. And that way you can look at those features again. So just in case you didn't get the right shot, just in case you weren't sure on exactly how many stamens or pistils were in that flower, you can now go and look back at that. So basically herbariums are the time machines to keep your research going. They also hold a tremendous amount of not only our knowledge of plants that are here, but also people who collected them. If you know anything about my talks, I absolutely love collectors. They're obsessive and I love them. I rely on them, and it's because of them that we have these tremendous collections here. And then this is just one of John Davidson's. He never made beautiful specimens, so this is the most beautiful one I can find. He tended to just take them, press them, and just keep going, right? So this is a beautiful specimen that he left us. So just to go back, so we have 6 collections in the Beaty, but we actually have 5 collections in the herbarium. So within the herbarium, we have 5 collections, and as Kashava was saying, we have 4 plants and 1 animal. So the idea with Linnaeus is that if it was growing out of the ground or seemingly to grow out of the ground, it must be a plant. So this is why algae and bryophytes and lichens and vascular, these are things that are standing still. They do not move. They don't seem to move from what we see, and they're emerging from the ground. So the idea was that obviously a fungi would also be a plant, and this is what it was assumed for a long time. But just by the way, that has now changed, and we now understand from a lot of the research that has been going on that actually fungi are more closely related to humans than they are to plants. So we keep them in our collection. I'm so glad because I absolutely love fungi and I wouldn't want to lose them to the animal collection, but they are an animal now, and so we do keep them on our collection. And mainly that's because they're consumers. A big reason is they consume, they digest, right? They don't autotroph, so they don't make their own food. So most plants make their own food, right? So they photosynthesize to make that food. So that's a real fundamental difference on theirs. But those are our 5 collections: the algae, bryophyte, vascular, lichens, and fungi. So just another shout out to some of these collectors, because I really want to encourage people to really start looking. So here we have we are the biggest BC macro fungi collection in BC's and in Canada, and we have 33,000, and 27% of those are just a Lunachesca. So I just want you to know that name if you love mushrooms. Lunachesca has done a tremendous job of collecting and has really grown our collection over the years. Another one is, of course, Paul Kroger. He donates to us often. He is just known for just having his eye out for new things. And there's a great story that happens to be on Beaty Treasures. So if you have never visited Beaty Treasures, we put this together for our 100-year anniversary a few years ago, where we picked 100 specimens that we thought were really cool. And this is one of them. And so if you read the story that Paul Kroger wrote, it's all about how this mushroom you see right here is actually on a textile. So he actually saw it growing on a couch, and this is a brand new species to science. So it's as easy as looking at a rotting couch, and you too could find a brand new species to science. The other shout out goes to, of course, Curtis and Trevor. They have built our amazing lichen collections. Most people walk by lichens, they might take a picture because they think they're kind of cool, they're kind of pretty, they've got beautiful colors, but most people don't understand lichens, especially at the level that Curtis and Trevor do. The cool things about lichens is that not only are they an algae, but they're also a fungi that have come together to actually make this brand new organism called a lichen. And there's actually new evidence that shows that there might be a third organism in there, which is a yeast. So we still wait to hear more about that, but they're incredible, incredible researchers who happen to live in their own beautiful backyard of Clearwater, BC. And I always have to do a shout out for Wilf Schofield because he built our bryophyte collection. He was an amazing collector. He collected so many, and his last collection number was 128,000. So when you're starting from one, just imagine you got a good 40, 50 years to get up to that 128,000. And there's nothing I can say more about appreciation for this obsessive, amazing collector, because our bryophyte collection is tremendously useful because of his work. And then I do another shout out for, of course, macroalgae. So this is my collection, and I've got 94,000 algae specimens, and they're just tremendous diversity. And we are ever growing an understanding about algae diversity with our amazing researchers at UBC. So we've got these 94,000 algae specimens, and 15,000 of them are more like the wine jar. So we've got these coralline algae that love to encrust and cover anything that it can't possibly can. And again, they're photosynthesizing. So there you have these beautiful pink colors, but this is all photosynthesis, right? And then of course, I wanted to show you this beautiful bull kelp, because bull kelp is an amazing annual. And you know, when you're on land, you would never think of an annual that could grow 35 meters, but if you're in water, why not, right? You're surrounded by nutrients, you need to get to that light. And so they can grow 35 meters in just 1 year. So it's about averaging 25 centimeters a day. But what I found so cute, and we made this into one of our fundraisers where you can actually buy these prints that we made, is that this is the hold fast, the whole bit. And so that's a very, very tiny little baby bull kelp. And so I just think it's adorable considering how large it gets later on. And again, I have to do this shout out to my obsessive collectors, and that's Frank Lomer and Jamie Finneman and Curtis Bjork. I have many more who donate to me often, but these are people who just go out on their own time, they collect, and yeah, you're gonna notice that Curtis is here again. So not only is Curtis absolutely an expert and amazing at lichens, he also happens to be amazing at vascular plants as well. I'm not sure he sleeps, but I love these obsessive people. They go around constantly looking for new things and finding new things all around the province that, again, we always think, oh, we know everything, we've seen everything. Well, actually, we haven't. And as we know, over time, things move and biodiversity moves. And these are the people who are tracking this biodiversity changes for us. So I just wanted to give you a little hint on where I came from, because I've been in this collection for 15 years. And this is what it used to look like. So it was really crammed. These were wooden shelves. They were not sealed. We actually had leaks every once in a while in our herbarium, and they were overstuffed. And then all of a sudden we got this amazingly beautiful space in the Beaty Biodiversity Museum. Our collection is in so much better care. I know it's going to last a lot longer now because the preservation is so much easier to take care of. And all I got to say is every time I look at this picture, I can't wait to get back to work. I'm hearing rumors we're going to be going back in the next couple weeks, very limited, but I just can't wait to go back into my space again and see my collection. I miss it dearly. Okay, so we're going to get into the bits now that we're here for, right? We want to learn how to press specimens, and we want to learn some about our spring plants. So I wanted to show you this particular picture. Again, this is one of these pictures that is on that 100 Treasures of Beaty on that website I was telling you about earlier. And so when we went to go look and find interesting things to put on this website for our 100-year anniversary, I really wanted to do something for John Davidson. And so I went through the BC archives, actually the UBC archives, and I went through the archives looking through a lot of his photos. So not only was he an avid collector, he was an avid photographer, and he enjoyed cycling very much. And so bicycling was a big deal back then, and he was avid cycler. So he would bike around and take pictures and collect plants. And I wanted to show you a picture that he took it's just great because he was trying to do what I'm doing now, is he was trying to encourage people to go out and collect. And so this was his collecting material. So you can see that he has his trowel, and he has a couple of snips, and he has a couple of tweezers, and he has his hand lens. And then he also has his herbarium press. And then you're going to see this very tall metal piece right in the center, and that's called a vasculum. So that's an old-fashioned Ziploc bag. So what we use now is a plastic bag. Well, he used a vasculum. So that's a metal piece. It's with a strap, and you just strap it around your body, and you would just throw your plants in there and then bring them back for the day to actually press. But one of the things I wanted to show you, which was so incredible, is when I was looking through these pictures, the specimen that's in the left-hand side, I saw it and I went, I wonder if we still have it, because this was from 110 years ago. And I thought, oh, you know, there's so many things that can happen to collections. And I was so worried. And then I went to the collection and there it was. And it looks almost exactly preserved, exactly the way he did it 110 years ago. So to have that experience, to see what taking care of a specimen can do, and knowing that we still have this specimen around 110 years later, is just a fabulous feeling for a collection curator. So when you're collecting specimens, the first thing you want to think about is really what makes a good specimen. And really what makes a good specimen is the reproductive parts. And the reproductive parts are the flowers and the fruits and the seeds. These are incredibly important. This is how we taxonomically tease things apart. It's the floristics that really tell us what plant you are and whether or not you're speciating, whether or not we have a new species. And so being able to get those flowers and those fruits and those seeds are very important. But of course, you're going to want to bring your leaves and your stems along the way, because actually, when you really start to get down to, in our world, when we start dealing with subspecies and varieties, you really often need to see those hairs that are along the stems and the leaves, because that's often an indication of a brand new speciation event occurring, where they're going to finally start to separate as species and become their own distinct species. The other thing I do is I check my field guide. And so you want to do things like check your field guide. I'm going to show you. So this is the one that I would suggest is the Pojar and McKinnon. The plants of the Pacific, the Northwest coast. As you can see, my book, I've had for a very long time, to the point that it is now come apart, right? I actually have 4 of these, because I have one in each car, I have one in my backpack, and then I have one that I've had forever, right? I've got all my little tags and notes of things I've been looking at. But what I do is I often go in here, I'll try to look at what I think the species is, and then I'll see if they give me an idea of how you would need to distinguish it from another species. So often you want to look at that just when you're going to take a specimen, just in case you might need flower and fruit. So not just the flower, but you need both sometimes to be able to identify. Or sometimes you need basal leaves, right? So the leaves that are at the base of that plant, sometimes you need those leaves so that you can really start to identify, because that's what's distinguishing it. The other thing is the roots. So often I don't take roots because I really want to leave the roots to be able to basically flourish next year for another plant. But sometimes you have to have the roots, because you have to define whether or not it's an annual or a biennial or a perennial plant. So there are times you want to do that, but I do discourage trying to take the roots unless they're needed, because it does mean that we want to encourage more plant growth next year. Now, before you go out, I've been encouraging people to collect in their backyards and along the roadsides around town, because often these are not needed for permitting. Most of the other places you're going to go, like the provincial and national parks, you are going to need a permit. And I always encourage, please get permits. It's important for these parks to know the diversity that they have. So whenever I collect and I have a permit, I make sure to get those lists to them so that they know what I've collected and they know what's actually in their park, so that they can keep a biodiversity list of their own parks. And then we also have the 1 in 20 rule. So when you go out, sometimes we don't know what we're about to collect. So it's kind of difficult to say, oh, I want to collect this, but I'm just not sure. So I'm not sure if it's rare, or I'm not sure if it's extinct or going extinct, not extinct, but going extinct if it's threatened, right? So we have kind of the 1 in 20 rules. If you see 1 in 20, most likely it's a healthy population. So you can take 1 out of the 20, right? So we've got this 1 in 20 rule about just taking 1 individual for every 20 blooming. You also sometimes, you're not going to want to collect sometimes. I have those moments where I'm not really sure, or maybe I don't have enough, I don't have 20 blooming. So what I'll do is I'll take a picture and then I'll take a GPS and then I can go back. And so that way I can come back to my herbarium, maybe talk to a couple of plant experts and see whether or not they've seen this plant before, if it might be new, and then we could actually go back and recollect it. But that way I have a picture of it and I have a GPS point, so that way I can go back if needed. Now, of course, if it's exotic, I always say take all you want. If you really want to control exotics, this is a great way to do it, is to basically harvest them, eat them, get rid of them, press them. But it's also great because then we can start to see how they're expanding their range, right? And we can start to see expansions. The other thing you can do when you take that photo is iNaturalist. So when you take that photo, if you're curious what it is and you're struggling with what it is, if you put it on iNaturalist, many times experts will come onto iNaturalist and help you out and get those identified for you. So the collecting materials, you're going to see here, I bring my gloves, I bring my secateurs, I bring what I call a hori-hori, so it's a digging tool. And then I have a little knife just in case I need to carve something off. I bring my binoculars because, hey, why not bird while you're out collecting plants? But also, what's nice about binoculars is if you're really stuck and you don't have a hand lens, you can actually turn the binoculars around and it becomes a little bit of a microscope. So it can be helpful to see small features, especially when you're somebody like me who doesn't see as well as I used to. And then I bring a couple of bags so I can put my specimens in to come back. I usually don't press in the field. It's very difficult to press in the field, of course, unless I'm out for the day, and then I'll start pressing out in the field. But mostly I'm going to bring them home and I'm going to press them once I'm home. So this is a lot of information, but I want you to just be able to refer back to this for later. And so when you're taking good field notes, you're going to want to take a good shot at what you think the genus or species is. And we happen to call that the specific epithet. And then in location, you want to put down country, province, and where you've been collecting. So the location of where you're collecting. And it's not good to put things like gas station on the corner of. Gas stations move. As we know, we barely have any in Vancouver as it is. But you have to really think in the long term. Where are you? And is this place going to stay that name for the next 100 years or 450 years, right? You're going to want to try to take a latitude and a longitude if you can, because that again gets you back to the exact spot just in case features change. As simple as, let's say Mount St. Helens explodes, obviously that topography has changed. So having that GPS point is really helpful. Most of us have that on our cell phone, and taking the elevation, which of course near here is going to be almost always about sea level. You're going to want to give us the habitat. Was it a street corner? Was it your backyard? Is it a forested area? Did you see any species around that were kind of associated, whether it was like Douglas fir trees? And then of course you want to put the collector. So you are the collector, and we want your full name because we want you for history. A lot of us have names that are quite similar, so it's really nice to give your full name so we can always give you credit for actually taking these specimens. And then you have the date. And I always want to warn people to do a 3-letter acronym for your month, because the United States and Canada does switch months and days. And for somebody like me who's trying to figure out what month or day that you collected to put that information in my database, it's really clear if you just put it as a 3-letter acronym. And then of course you always want to use all 4 numbers for the year, because I have many people who have entered data that has put something like 2019 when it's 1919 or 1819. So we do want to make sure to put all 4 of those numbers in. And then of course we want you to put down if there's anything interesting about the plant that you collected. So the color, the smell of the flower. Remember, a lot of flowers lose their smell. They also lose their color. So sometimes again, it's nice to take a picture and also describe what you're seeing. Here's an example of Frank Lomer's. And I bring this particular specimen up. Oops, sorry, let's go back. I bring this particular specimen up, and it's mainly for the notes. So if you look on notes, he says several plants in one area established for many, sorry, my zoom's in the way, established for many years, only known site in BC, native to Europe. So basically what he's telling me as a curator is that this is an invasive plant, and this is one of the only places that he has found this plant, and it's something that needs to be watched. So he collected this in 2008, and so it'd be interesting to see whether or not this is overwintered and dispersed. But this is the kind of information that these collectors are giving us. in their note section is these kind of invasive moments and whether or not they're spreading. This is another one that's from John Davidson. And so you'll see again down here, the date is 11/7/12. So that's 1912, not 2012. And this is where it's again important, was this November or was this July, right? So this is where it's important to put those 3-letter acronyms. This was on Hollyburn Ridge in North Vancouver, collected in 1912. And this is one of the notes I really love. So this is another note that he had, and this was important for herbariums, is we had these collections because a lot of people were trying to figure out what they could eat, what they could harvest, and what they couldn't. And so this note particularly tells you that you're not going to want to eat this because it's proved fatal to man and horses. And there's some really nasty symptoms that can come along with this, including increased salivation, producing a weak pulse, labored respiration, and profound prostration, which of course would mean you're going to be throwing that right back up. So it's a good note to have that I obviously do not want to eat this plant and I want to avoid it at all costs. So with the pressing, we got a simple and free pressing materials around your house. So you have your cardboard pieces. Everybody's got cardboard around their house right now. We've all been getting everything shipped to our houses. So you're going to want to make them 12/17, and you should see the C's along the long edge. So this is going to be the edge for drying, and this makes the ventilators, right? So this is the ventilation. And so you're going to want the long edge to be able to see those C's, right? And then you're going to use newsprint. It's free. It's around town. Georgia Straight is great. Just don't use those glossy sides because they're not going to really absorb the water that you want. And then you're going to put on a weight. So the best thing, the cheapest thing is, of course, a big old book. So I've got a big old atlas of the world, and I've got my cardboard, and I've got my newsprint. So those are the 3 things you need. Is all you have to do is put your pressed plant in the newsprint and inside the cardboard, and then put a nice big book on it. It's nice, consistent weight. That's what you're looking for, is consistent weight to hold that plant down. And as it's drying, and then I usually make a little tag for the plant for the inside to tag it, just to make sure that I've got double information on what I've collected and where I've collected it from. So these are not free, but this is what you're usually going to see. So this is my own personal press, and with the straps, and then I've got my cardboard. I also sometimes use foam, depending on what I'm pressing and how I'm pressing it, but the foam can really help to level, again, when you've got a very uneven press. And then the last thing I have on top there is blotter paper, and blotter paper is really important for absorbing. It's quite expensive though. So I think it's like a $2 a sheet. So it's a very expensive, but it's very absorbent, and it also lasts a very long time. So what you're trying to do now is you're going to go and press your specimen, and you're basically just trying to get the water out of there. You're trying to extricate out that water. And so it's going to take usually 3 to 5 days. It's dependent so much on the weather. It's dependent on how thick the specimen is, whether or not it was spring. So there's a lot of factors that can play a part in whether or not you have success or not success with your with how fast it's going to dry. But the most important thing is when you're pressing and you've got that newsprint, you're going to want to change that out within 2 days of doing it. You're going to want to check the moisture. What you don't want is you don't want mold building up, and it's really about getting that newsprint and constantly keeping it dry. So it's about consistent drying. If you have a plant, and let's say you decide to put it, like if you live in a basement apartment, often those can be quite moist. And so to keep that dry is quite difficult. And so it'll keep reabsorbing water from the atmosphere, and it'll cause it to go wrinkly or not quite be the flat specimen that you were hoping for.
Linda, can I jump in with 2 questions?
You bet.
So just going back a little bit earlier to collection, Nicole's asking, do you ever worry about GPS or location data for rare plants like orchids, as in the information becoming publicly available?
So we are concerned about that, but as a collector, you're going to want to take that information and we're going to want to house that information because otherwise it's really not a great specimen if we can't figure out where it came from so that we can map it for the distribution. But what we can do is we can hide that information from the general public. So again, that information is important for you to relate to me and to give to me as the holder of that information. And then I can share that information with places like the Conservation Data Center of British Columbia. But if we really feel like it's something that needs to be hidden, we will hide that information, and only can somebody request it that we feel comfortable with releasing that information to.
Awesome. Just one more quick question. And I did want to acknowledge that there were a lot of comments earlier, a lot of people sharing their favorite plants. So the other question is that Sheila asks, where locally can we purchase blotter mount paper?
Yeah, so it's difficult locally. So just so everybody knows, I normally sell plant presses. I have an amazing volunteer, Andrew, and he's in great construction, and he is the nicest person ever. And he started making me plant presses to sell to the general public just for fundraising. And so I have plant presses that are in our building that I would love to distribute to you guys, but I don't have access to them. Now you can find them online, but I do find them to be quite expensive. Understandable, but I was able to offer them for a lot less because I really wanted to encourage people to get out and not have that be the issue. Blotter paper, again, I get it from the United States. I have a particular herbarium company, herbarium supply company. I've been with him for, actually, our place has been with him for 30 years. He's an amazing supplier. You can get blotter paper at archival companies. McLaren, I believe, sometimes has blotter paper. They were out recently and they were out of their plant presses when I looked recently for somebody. So the supply is difficult. If you don't have blotter paper, you can use paper towels. Right? Paper towels are absorbent. That's what they're supposed to do. So really, if you want great color in your flowers, you want great color in your specimens, and you can't get a hold of blotter paper, just use a paper towel. Just put it in there with your specimen and just make sure you're trying to keep it absorbing and not getting more humidity back into that plant.
Awesome. Sorry, there's one more quick question. And I did want to say, it looks like Andrew is here and he said, aw shucks, when you mentioned him. So that's sweet.
Yeah.
And just one more question for now. Deepti is asking, do the plants lose color if we open frequently to check for mold?
No, it's the drying. So you can open them as much as you want. That's actually what you want to do. So when I actually go out in the field, I have 16-hour days. Don't tell my boss. And when we do that, it's because I get out early, I get the specimens, I collect, I then press them. And then I want to smash them because I want them to start doing what I want them to do. And then I go and I open them back up later in that day and I rearrange them with like little tools to make sure that everything's open the way that I want. And then I'm going to close it back up. I'm going to let it dry for about 2 days. I'm going to open it back up, make sure it's still staying moist. If I need to put paper towels in there or better blotter paper or switch out the newsprint, I will do that. And then I'll close it back up. What's going to affect it is if you can't get it to dry, you're going to start having mold issues, or you're going to lose the color if you can't get it to dry quite quickly, like within 5 days. So you want to try to, again, get it as dry as you can, but don't overheat it, right? You don't want to overheat it. That will absolutely take out all the color and will destroy the DNA that we can use.
Awesome. Okay, that's it for now.
Cool. So identifying specimens is almost a whole nother Beaty at home. We've got 250,000 plants. That's just vascular plants in the world. I'm not talking about algae, bryophytes, fungi, just 250,000 seeding plants in the world. That's a lot of plants to try to learn their names, and they're constantly changing names. So if you do track this stuff, they are constantly changing names. And it is valid. We are finding that we did not necessarily understand everything through the morphology that genetics is allowing us to do, just like we've seen with fungi, how we now understand that it's much more closely related to animals. But I did want to show you what I did have on my bookshelf, at least at home, when we went into COVID. And the two that you're going to see that have my little tickets in there, again, is Hitchcock here. This is Pojar McKinnon. And that's the book I go to often just to get a quick reference. But then if I'm going to key it out, this is the brand new Flora of the Pacific Northwest. So this was the old Flora of the Pacific Northwest from the 1960s. They just updated it this year. And if you're somebody who is really into knowing your species and really getting it down to variety and subspecies, this is an excellent, excellent book. And I'm really, really been happy with it so far. It's very manageable for keen. And then I have other things like if you do, if you're a pond person and you like your water species, I've got the ponds. I'm trying to read sideways. The Pond Burrweeds and Their Relatives of BC, and that was written by Brayshaw. So you can get these really specific books as well. So if you're somebody who is really into specific things, you can actually find those books as well. And then the online. So there's a lot of online resources, but of course, if we're going to start off, we're just going to go to eFlora BC. They've got great pictures. They're vetted. So don't go to Google, okay? Those are not vetted by experts. Every single photo that goes into eFlora BC is vetted by an expert. So they're not going to have something up unless somebody has cleared it as correct. And then you're also going to be able to look at distribution maps. This is incredibly important. So if I'm stuck between a species and I'm not sure if I have this variety or this variety, often I'll go and I'll look at the distribution and just make sure, could it even be in the area I'm in, right? So has there ever been a collection before? So often it can kind of help me tease out whether or not I've actually got the right species, just based on their distribution. There's a lot of other helpful information that's on eFlora as well. So for mounting, we just got materials. I use archival mounting paper. Again, I get it at the Herbarium Supply Company. I get also my archival pH glue balanced so that it doesn't react to the plant. And I used gum linen tape. And so this is to ensure the long-term preservation of the specimen. So what's weird about scientific specimens is that you don't want to use a lot of glue because we actually often need to lift that specimen off the paper and then look at it again. And so if we glue it, we can't easily remove that. And so if we just use this lignin tape, then we can actually cut and lift those specimens again. When you're mounting, you want to make sure you're not going to mount to the edge. So right here, this is a beautiful specimen. It's absolutely gorgeous, but I would have flipped that flower and put it in the center. It's the most important piece there is those two flower pieces, those reproductive pieces. And so you really want to flip those and put them back into your center because the center of the paper is the safest place. Once you get it to the edge, that's where it's most likely going to break off, right? So you want to kind of keep it to the center. You want to keep those reproductive parts around so that you can really see the reproductive parts, and hopefully those will last a very long time. And then you're going to leave room in the lower right-hand corner for, of course, when you make your label, whether you type it, handwrite it, we make labels for you if you donate, and you will put that in the lower right-hand corner. So you leave a little bit of room. And then how to mount a good specimen and how to save a bad one. So this is a nice specimen, and then you can see the pieces of tape that are across the reproductive structures here. But what I will say is right here, you can see tape possibly on the edge of each one of these tips, and I would never do that. So that's going to break, right? So you're going to put that tape somewhere else, or you might put a little bit of glue behind. It takes years to understand how to mount these properly. Each plant has got their own personality, and this is what I often talk about with my own mounters. I have volunteer mounters who come in once a week normally, and it's about 15 of them, and we sit down and we learn techniques on every single plant. They all have their personalities, they all have their breaking points, and it's trying to make sure that we're going to press these but also mount them so that they're going to last a very long time. And if you have extra bits, as you saw on Instagram possibly last week, you can just take those extra bits if they didn't work out, and then you can start to make crafts. I often make my Christmas cards, birthday cards with all the extra bits that I have left over. So nothing really ever goes to waste.
Linda, can I just quickly jump in?
Yeah.
A question. So Sheila asks, do you know if Ride in the Rain field log paper would work for mounting?
No, it's not absorbent. So the whole point between about Write in the Rain books is that you can write in the rain, but they actually don't absorb water. So it's actually going to push away water rather than absorb it. So I wouldn't use Write in the Rain books. I would for writing on, but not necessarily for using anything for pressing.
Awesome. Linda, just letting you know, it's 1:40 now, just to keep you just on time. Okay. And then I just wanted to let everyone in the chat know as well that Nicole is sharing all the links that Linda is mentioning. So if you'd like a clickable link, it's in the chat.
I didn't mean to talk this long. So last one, I hope, and then I'm going to just show you something, a couple of specimens and a couple of things in the last hopefully 10, 15 minutes. The last thing is a lot of people forget this part. You got to freeze your specimens. So all the insects go and they deposit all their beautiful little eggs and their little babies inside of these flowers right now. And so the problem is when you go to press them, you might get rid of all the big bugs, but the problem is all these little babies are still there. And if you do not freeze them, then they are going to emerge and then they're going to eat your whole collection up. They absolutely love the pollen. They absolutely love seeds. So they're going to eat it right up. So just wrap it up in a plastic bag, tape it up, and then put it in your freezer for a couple of weeks. And then you have to repeat this often. Dermestids are what's going to go after there. And so that those little insects are just going to eat away at that pollen. And then you're going to want to store them flat in a dry, cool place where there's no exposure to light, because it's the light that's going to wreck them. So that's it for that little longer talk than I thought. And again, you can always email me questions, and then you can follow the Instagram for more stuff. All summer, I'm going to be doing the backyard biodiversity, trying to give you more hints on different techniques, and also doing some of that backyard biodiversity about some of the plants that you might be finding around town as you go walking about and in your own backyard. So now let's see. I want to see if this will work. So what I want to do is I'm going to switch my computer. Yay. Okay. So for the end part here, there's never enough time to show you all the things. I also just wanted to show you real quick. This is a little press that I made just out of cardboard and some strapping. So that way I could just throw it into my little pack if I wanted to. And that way I could just get little things. There's lots of little plants that are around town. And so I love little plants. Most people tell you if it's like below, you know, a couple centimeters, I totally want to grab it. So, so that's a little press that you can make yourself. But a lot of people start asking me, okay, how do we do this, right? So here is my press, right? This is the base. And I've got my straps here. And everybody can see that okay, correct?
Yes, Linda, looks good.
Right on. So the first thing I'm going to do is I'm going to put down one piece of the cardboard, right? And when I have, that's your long end, that's your C's, right? So this is your long end. You're going to lay that down flat. And then you're going to take your Georgia straight or newsprint that's roughly the size of what a plant specimen would be. And then you're going to take your specimen.
Hi, Linda. So when you're a little further away, we can't really hear you.
Okay.
So we didn't catch that last part.
So then you're going to take your plant and you're going to press it. So here's one of the ones I wanted to bring to your attention. So a lot of us know this is salmonberry. I just this morning on after my little walk with my dogs. And right now, as you can see, this is a lot of material, right? We're not going to want all that material. If you try to press all that, that's not going to make a good scientific specimen. But one of the reasons I wanted to bring this up is because I wanted to show you kind of how people will ask me about berries and how to deal with that. So what I'm going to do is I'm going to take my secateurs and I'm going to just cut these pieces away. Basically, I'm going to give it a haircut. What we're all dreaming of nowadays, right? Haircuts. And in all honesty, I want to, for this particular specimen, I want to show off those berries because I want to show off that color. And often what I want to tell you guys is it's great to have those flowers, but I would love to see more collections where people are collecting the fruits. We don't have enough of those in our collection because so many people do go after those flowers. And when you have your berries, or sorry, in these particular case, these are not berries, these are aggregate fruits. So each one of those is a little fruit in itself. And so this is an aggregate fruit, which just means a whole bunch of fruits in together. And so what I'm doing right now is I'm taking wax paper or parchment paper, and I'm going to slip it underneath and on top, and then I'm going to squish it, right? And the reason I'm going to squish it is because one, I don't want it to mold in my newsprint. I don't want it to mess up the rest of my specimen, but two, I need to see the seeds inside. So I need to see if there's 1, 2, or 3 seeds. So you often want to do at least a couple of them where you're going to squish it with that parchment paper. So anytime you have anything sticky, right, you're going to take that parchment paper, and again, you're just going to take it and you can squish it and then keep that when you're drying, right? So it doesn't mold the rest of the plant, right? So you're going to do that kind of thing where you have these. This is protecting, but now I can taxonomically identify this because I can now see how many seeds are inside. And then I'm going to take my little, this is a little ticket that I made earlier. right? So I'm going to do my Rubus spectabilis on June 17th. I collected it, and this is my 15th collection of this year. And I'm just going to put it here as a tag, right? So I can do that and put that as a tag. Now, if I have the time, I would sit here with you guys and I'd really arrange this so that I don't have overlapping leaves like I do here. What I want to be able to see is the characters of these 3 leaves. That's what defines this group. And then you spend time. And then this is what I mean is now I've got this shrub and it wants to fight me. And so I have to then tell it what to do. So I'm going to snip that part out that's just causing me trouble. And I'm just going to lay this flat. And then I'm going to take the specimen, cover it, and then you're going to hear my crunching. And I'm going to get this plant to do what I want. And then I'm going to take another piece of cardboard and I'm going to squish it. And that's what I'm going to do is just put it in that as much as I can to try to basically break some of those cells so that it will start to relax. And this is why I revisit it later today, is I want to check and see if it's relaxed and if I need to move anything around to make it a better specimen. And then what you do is you do your next one. So then you have your next plant. This is another one that's coming out right now. This is Salao or Gaultheria Chalon. This has very, very sticky ends, right? So this is another one where I don't have to squish it, but it will stick to your newsprint. And so the problem is when you go to actually, after this is pressed, you're going to get kind of frustrated because often these flowers, because it's such a sticky end, they will actually stick to your newsprint and it'll wreck it. So again, with this, I'm going to use this parchment paper or wax paper to cover it to protect it. Now, it's not going to dry as easily in here, so I will have to check this to make sure that it's not molding. But this is another way that you can deal with anything sticky. Also, if you're somebody who likes to collect things with bulbs, bulbs are quite sticky. And then again, I'm just going to squish it and crush those cells, make them do what I want. And then I just start to layer, right? Now, if I have a really wet plant, which sometimes I do, that's where I bring in the blotter paper and I'll put the blotter paper on both sides. Which, so if I've got a really wet plant, I will put the blotter paper first. Then I will take the newsprint, put it down, and then I will put my specimen. So let's say if this was just the specimen, then I'd put my specimen, lay it out really nicely. If this was a wet specimen, cover it, then put my second piece of blotter paper, and then my cardboard. Just like that. So all you're basically doing is a layered cake of dead plants that you've cut and now you're going to crush and now you're going to make into beautiful, beautiful scientific specimens. And that's all you keep doing. So you just keep layering and layering. And then at the very end, you're going to take your press. You're going to put the top piece on. And then you're just going to strap it closed. And again, putting pressure on, depending on what's in here. If you've got something that's a lot of shrubs, you're going to need to pull that down tighter. If it's something that's quite gentle, more delicate, you don't want to pull it down too tight because you can actually crush the flowers. And if you notice, I'm also doing it in the opposite direction. So you're going to have tension in both directions, one in one direction, one in the other. And then there's my press, and that's my pressing for the day, right? Your press can be as tall as your straps. And that's kind of what blew me away about when I first started pressing. A lot of people think when you press, you know, it's going to only be that tall. Your press can be, I've had my press as tall as my hip, so almost about 2 feet to my hip when I went out and did a bunch of pressings for a project. So your press is just dependent on how long your straps are. So if that gives you a little bit of an idea, I'm not sure how much time we have. I can always show you a few more, but I'm not sure how much time we have, Kashaba.
It is 1:50. Possibly one more.
Okay. Hold on. One more. Okay, so it's always hard to do one more because I've got at least 4 or 5. But here's what I want to show you. So this is sword fern. Okay, this is incredibly long. A lot of people again ask me, how do you deal with something that is longer than the actual sheet itself? So 2 things is you can actually just cut and make it where that's the first piece, then this would be the second piece, and you can actually piece it back together. So you'd have 1, 2, 3, and again, you would layer each one, and then this would be my last piece. So you can actually put them all back together, or you can do it like this, where you bend it. And what you do is you do what's called a V or a Y or a W, and you just bend it like this. This is quite nice because then you can see the front of the leaf, plus you can see the back of the leaf, right? Which is quite nice when you have this kind of effect. If it's so tall, sometimes that won't work and you will need to cut it into those 3 pieces, right? So depending on what you're working with, and so you would just press it and then just close it up. Put your cardboard on top, right? Get them tucked back in there and squish them. And then you'd have your big, gigantic sword fern, right? And you could keep going. And things like, you know, this is another one that's out right now, is our beautiful Buttercup. This is unfortunately the invasive Buttercup, but this beautiful little Buttercup here. And so not only did I collect the flower and the seeds, but I've also collected here, I don't know if you can see this, but this is actually the extension. So this is showing that it's a runner. And so it'll actually take off and it'll root. And so that's another important feature to know, that this is a particular ranunculus that will actually make a runner. This is often in very wet environments. So this is one that I would use blotter paper with, because it's most likely going to mold if I can't get it dry fast enough, right? And so you would just arrange. You would have one leaf showing the colors here on the front, and then I'd have another leaf that I'd flip so that you could see the back, right? And so you always want to be able to do that. If it's too long, you can start to bring it in. And then often I'll open up the flower, push my thumb on there to get it to squish so I can see the inside. And then I would close it up, and then I'd revisit it later today and just make sure everybody's aligned properly.
Awesome.
Cool. What?
So Linda, we do have some questions. So whenever you're ready.
Sure.
There are just some comments like admiring your technique. Oh, hi. So Nicole's saying the squishing of the berries with a wax paper is a really good tip. Daniel's saying it looks so easy when you look at the finished product, but this is a great way of seeing the work and artistry that goes into those gorgeous specimens. Deepti asks, do we get balanced pressing with just 2 straps on the plant press, especially if you have a big specimen like bulbs?
So that's a great question. And so this is where the one thing I didn't show you guys is, so when I'm talking about when you are trying to press a lot of specimens, so right now I'm lucky, I'm going out and I may be doing, 5 here and there. But when I go out in the field, I've got to get as many as I can. So sometimes I'm getting up to 50 a day, and I'm going to be having shrubs, I'm going to be having perennials, I'm going to be having them all next to each other in the press, and they're going to be fighting each other in the press, and they're going to be weighing each other in different ways. And as much as the straps are supposed to even this out, what sometimes can happen is if you have a shrub that's right next to a really sweet perennial that's underneath it, it will crush it. And so what I'll do is we had a bunch of foam left over when we moved, and we started using it when we were making the pressings. And this actually helped weigh it out and actually level it. So this way, if I have a really beautiful, this very succulent feeling. So this is Siberica, this is Claytonia siberica, right? So this is Siberian miner's lettuce. This will just turn into almost water when you squish it. So I would put this in, obviously a lot of cleanup, and then I would put the foam on top. And then if I had a shrub like a willow, or if I had even salal, then I would put that on top, but I'd have this foam to be able to level it, right? So they're not affecting each other so aggressively. So there's a lot of different techniques that you can do depending on what you're pressing. You know, we haven't even gotten into if you're doing cactus, kind of nightmare stuff you gotta do when you press cactus. And this was only vascular. So, you know, algae is a whole nother fun issue that would be great to actually cover, because a lot of people are getting into actually pressing algae again, which is wonderful. But that's a great question, Deepti.
Awesome. And Deepti says, thanks, that's a genius solution. There's a two-part question from Nicole. Do you encourage like the front back of leaves, flowers, and fruits when possible? And if you have extra material like loose seeds and needles that fall off, what do you do with those?
Ooh, so I don't have that, but yes, that's a really good question. So since the most important part is these beautiful flowers and the reproductive parts, often, sometimes when I'm pressing, they're so clustered, they're in the way of each other. And I don't want to wreck the specimen, right? I don't want to press these all together to the point that you actually can't see anything. So every once in a while, you know, especially if you have time, I love time. When you have time, you can just pop one of these off, and then I would press it all by itself so you can see all the features. I would take these wonderful seed heads and do the same thing and just press them by themselves. And then what we do on our side when it comes to herbarium is we take these, and if we can lay them on and mount them, that's great. But if we can't, we put them in what's called a fragment packet. And a fragment packet's really wonderful because it means if another researcher comes in and wants to look at this seed head or look more closely at this flower, they do not have to touch the original specimen. They can just look in this fragment packet and have a little look and see these little bits, and that way they never have to wreck the actual big specimen, and they can just look at those little bits. So I will say in my job, nothing goes to waste. Like from foam to little bits of flowers, everything. There's hardly anything. We always reuse and recycle because we're constantly using anything we can, but also we don't want you to throw out these bits as well. And also remember, you guys, this is DNA, right? So even this leaf, if I clip it off, I might keep the whole thing because I know that I can even crush this and I can still get DNA. So even if I morphologically can't decide what this is anymore because it's been so torn apart, it's actually DNA that, again, I can use to extract and understand more about this biodiversity of the plant.
Awesome. Thank you, Linda. Christine has a really good question. When pressing something like a plant disease, such as a gall, yeah. Yes, how would you do it?
Yeah, so galls are interesting. So you do want to keep the gall. So on top of the fact that I'm collecting a plant specimen, in all reality, I'm also collecting tons of bugs, right? I always joke around, if you're vegan, you're not really vegan if you're doing this. And so you do want to keep those galls. And again, this kind of foam, even being able to cut off a little piece of foam and have that be able to put over that gall in the hopes that you don't want to crush it. Like, I didn't even get into the particulars of things like cones. You know, you don't press cones, right? You just dry them. But that gall is attached and it has information in it to biodiversity that was attracted or predatory on, predatory on these plants. And so you want to try to keep it intact. So just in case somebody who's studying that insect can come into our collection and look at that. So again, being able to use things like foam and not completely crushing it, but being able to hold that casing that they have is really nice.
Okay, I think there's some questions from Facebook, so Nancy will.
Okay, fantastic. Sonny wanted to share that he related to the collection notes with his second year herbarium collection for forestry. And he also was thinking about bird watching and how you don't usually disclose locations of rare species there. But we did have a question. So Tobin is asking, once we have dried our specimens, how do we donate to Beaty?
Oh, sure. You email me. So normally, like I've been doing workshops for the last couple of years, and I talk a lot about how to get a hold of me, but I'm on email. And honestly, like, it's you guys who are going to collect. It's your guys who are going to build our collections. And this is why I do these workshops. This is why I want to encourage you now. You know, you can always contact me. I can guide you, but there's information that you would want if you're going to donate to me. So including, like, I have an Excel spreadsheet for you to fill out with all your collection information. I can give you hints on what we would maybe need. But also, if you're finding anything interesting, I often want to hear, because often it's the people who are walking around seeing this cool stuff who's going to bring it to us, and then we can bring it to the attention of people like Curtis, Amy's of the world, right? And then they can go back and start to see whether or not this is actually a brand new species to BC or to science. So contact me. I can always help you get things donated. I work with many different people, and even if they give me one specimen, sometimes this is the first specimen that's ever been seen in BC, and it starts a whole new cascade of understanding of our biodiversity.
That's great. Thank you.
Yeah.
By the way, also Elaine says, wonderful to see you. Great presentation. There's some thank yous as well. Thanks.
Awesome. So do we have any last questions? We have lots of thank yous here on Zoom as well. A thank you from Joe. Thank you from Jasna. Thank you from Christine. Oh, Valerie says, when I grow up, I want to be like you. That was awesome. Thanks, Linda.
No, I really miss you guys. And I really hope for all of you who maybe have not seen the herbarium or come to the museum, we want you here, but also while we're away from each other, I'm on email. And, you know, if you've got a question, if you're wondering a species, you know, there's a lot we didn't get to cover today, right? There's not enough time ever, as you guys know. And there's a lot of different plants out there to collect. So definitely just contact me. I'm up for helping anybody and everybody who wants to start learning how to collect those specimens.
Awesome. If there are no more questions, we might wrap up there. One comment that I missed was Deepti said, would love to learn how to press fungi maybe in the future. Do you do that, Linda?
You don't press fungi.
Okay.
You don't press fungi.
Oh, that's right.
So I'm trying to we have a brand new employee. I love her, Karen Galinsky. I can't wait. But she is new. She's new in COVID. So imagine starting a brand new job during COVID. But she actually is going to be taking care of the mushroom collection and the lichens and the bryophytes, and none of those you press. Those are the coolest because you just grab them, dry them, and then you put them in a little packet. So they're a lot less work other than identifying. That's a little harder.
Okay, awesome. So lots of thank yous. I think we'll wrap up here if that's okay. And I just wanted to say thank you to everybody for joining us for another wonderful Beaty at Home. Thank you, Linda. This was so excellent and so great to see all the plant pressing. Just wanted to let everyone know we do this every Wednesday at 1 p.m. And next week, we're going to do an experiment along for families and anybody above the age of 4 or even younger. And we're going to specifically do a little chromatography experiment. So it should be lots of fun.
Cool.
I'd like to join. Yeah. Okay. So thank you guys. We've got more thank yous rolling in.
Thank you guys so much. Honestly, I'm so excited. It's been such a great summer. The spring has been amazing. The rain, I mean, I don't love it, but my gosh, the flowers are incredible this year. So, you know, embrace it, right?
They are awesome. Okay.
Hi, Amber.
Oh, okay, great. You can see the chat as well. Excellent. There's a thank you from Nate, who's 9 years old.
Oh, hey, Nate. Thanks. My 10-year-old is very bored at home.
Aw.
Oh.
Are we still streaming to Facebook?
Okay, you're done wrapping up. Okay, great. We will turn that off then. Okay.
Awesome. Thank you.